Hello and welcome once again, my dear students, in this ICSE biology series. So, since we had already started with chapter number 1, cell division, जो हमने कर दिया है, cell division हमें करना है, cell we have already completed, children. And before I would begin with cell division, what I wanted to explain certain terms so that I could take up the chapter of cell division, and at the same time also be confident enough to take up the chapter genetics. So in the same process, let me do the slideshow. In the same process, what I was trying to explain you certain terminologies. Those terminologies include chromosomes, chromatid, chromatin, fiber, and centromere, and so on. And not to confuse it with terms such as centrosome and chromosomes. All these appear to be like similar terms, like for once a child can get confused if he is studying them for the first time. So therefore I thought that it is wise enough for me as a teacher to help you explain and get used to these terms, and so that we will, when we are using them frequently, when you're talking about very quickly, if I go on speaking and I start talking as chromosomes, centromere, chromatin, the child does not get confused between these words, and he has a very good clarity as to what is being meant when these similar sounding words are being spoken at the same time, which we would be using when we are doing the chapter genetics and when we are doing cell division. So in the same process, I was on this slide, which gives you a picture of a chromosome. Now this particular picture of a chromosome, which child you can see in front of you, this is one chromosome, as you can see. So this one chromosome has actually begun to compress itself or condense itself. It's in the cell division process, and it has begun to condense itself, which is why we are able to see it in the form of a rod shape. So yes, we can see it in this rod shape because it has condensed. Okay, it's ready to go into cell division. So these are the arms of the chromosomes, which I can say as P arm and Q arm, which are held in place or which are joined by the structure which is called as centromere. Do not get confused centromere with centrosome. Centrosome are what? These are the cell organelles which are found in animal bodies, animal cells which give rise to spindle fibers during cell division. And centromere are the bodies or the condensed structures of regions inside the chromosome where the arms of the chromosome get attached to themselves. So this is one chromosome, as you can see, and these form the arms. Now here we see, still I have in the second diagram, children, I'm talking about the second diagram, you see here again one centromere. However, now we, instead of only P and Q arm, we have got two P arms and two Q arms. So if I say this P, then this is also P, and this is Q, and this is Q. So we have two P arms and two Q arms. So what has happened is that the DNA content inside them has got duplicated or replicated. You see the word here, one chromosome after replication. So what has happened here, child? That the DNA content has duplicated. So if the DNA present in this one chromosome was supposed to see here, the DNA here would be 4C, because we have got two arms of P and two arms of Q, because it has got replicated itself. Yes. Now, in the process of replication, what do we see? We see two P arms and two Q arms. So that one half of the chromosome here, child, if I were to divide it into this form, then this one half is called one chromatid and the other half is called another chromatid. So this is one chromatid and this is another chromatid. This is one chromatid and this is another, the second chromatid. Now this chromatid you will be understanding more because I've got more pictures ahead in the coming slides. So this is one chromosome. Yes, this is also one chromosome and this is also one chromosome. So all the three diagrams show only one chromosome. But what is different in the second diagram? The second diagram shows two chromatids instead of one chromatid, which is seen in this one chromosome and the in the first diagram, and one chromosome which you see in the third diagram, you can see one chromatid per chromosome written. You can see here one chromatid per chromosome written. And what do you see here written? It is written 2 chromatids per chromosome. So as we move ahead, you will get more clarity. Let us just give a reading to what we have done so far. The first bullet talks that the chromosomes are thread-like. So can you see thread-like structures in the diagram here on the left, on the right, sorry. So this is, can you see this thread-like structure? So it is a thread. You can see it in a thread, like a thread, which has condensed itself, highly condensed itself so that it can undergo cell division. Okay, and what does it contain? It contains genetic information inside it. So this thread is containing genetic information which is going to be passed on from one generation to another. So it has a vital role in heredity, cell division, variation, mutation, repair, and of course regeneration. Then in the interphase, that is when the cell is getting ready to divide itself, what we can see it as a thread-like structures which will if I try to obtain, if I try to stain the nucleus at this particular stage, which stage? It is the interphase stage of the cell division. At this particular stage, if I try to color the nucleus, then I will see that there are certain structures within the nucleus which can take up the stains. So the stains that I would be using are acetocarmine and gypsa stains. Now, I'm not giving them in so much of detail because you don't have to learn it as of now. Simply just writing you down the name of the stain, which can be used to color or show the distinct parts of the nucleus to be observed under microscope. So there are certain structures which will take up the stains. Okay, so what those dark staining fibers which are taking up the stains are called as chromatin fibers. So they were named as chromatin fibers because they had the ability to take up the stain and get colored. Okay, colored, chroma is for color. So that is why since they were getting colored under the presence of a stain, so that is why they were called as chromosomes, which in the form of a thread are nothing but chromatin fibers. When they condense, they form chromosomes. When they are in a thread-like form, then they are as chromatin threads or chromatin reticulum or fibers. Now, as the cell begins to enter the first stage, that is after interphase, when the cell has entered into cell division, that is the first phase of cell division, which is also called as prophase, which we would be dealing in detail in the coming session. So what do we see? That the chromosome fiber has condensed to form chromosome. So this is what you see, child, in the interphase. In the form of telephone wires, you can see that the coiling has begun to take place. You saw it in the form of a thread, then you saw it in the form of a very high coiling structures like that present or seen in a chromosome, and then which will get further more and more condensation will take place, giving rise to the structure called as chromosome in the prophase of cell division. And we would be talking about these yellow things which you are seeing in this particular diagram in the coming slides. Now, what are they? I'll be I'll be talking about them. Now, so what is the purpose of this particular class is that we want to see that how this approximately 2.2 meters length of the DNA, the chromatin material that is present in each cell which has this much length of DNA inside it, how is it able to get condensed in a structure called cell which has the diameter of 10 to the power minus 6 meters or micrometers, in the range of micrometers, that is 10 to the power minus 6 or a million times smaller than a meter. So how am I going to pack 2.2 meters length of DNA? 2.2 meters length means it's even more than my length. If I take my length, then it's obviously it's less than 2.2 meters. So I have to coil a thread, or the cell has to coil a thread within it, within a diameter of less than a million times smaller than a meter, which is the size or diameter of a cell. So are they not such beautiful packers? Is not nature, or have nature has developed such a beautiful mechanism of condensation of this particular thread of length of 2.2 meters into as small a structure, which is as small as 1 by 1 millionth time smaller than a meter. So let us see how this kind of packaging takes place inside the cell. And just to tell you a fact that since I have already spoken that we are made up of how many 40 to 50 trillion cells. So we are made up of 40 to approximately 50 trillion cells, maybe around 10 to the power 12. So if I were to open up the DNA which is present in all of these cells that make up my body, that is 40 to 50 trillion cells, the length of the DNA, the total length of the DNA which I would be getting in all the cells, 40 to 50 trillion cells, that length would be equal to 300 times, rather more than 300 times the length between Sun and Earth. So isn't that quite phenomenal, children? So if I were to unwind all the DNA from all my cells, and that length is even more than 300 times the length between Sun and Earth. So that means what? Even the whole of the Earth cannot is less to have that much of length of DNA present inside it. So how are we going to package such huge lengths, that is 300 times the length between Sun and Earth, in one organism? And we'll see that how it happens within each cell. To help you explain that how packaging takes place, let us first try and understand what is the chromatin material or the chromosome made up of. So this chromatin material or the chromosome is made up of basically two things. First is an acidic material and the second is a basic material. The acidic material is DNA. The acidic material is the nucleic acid called DNA, which is called as deoxyribonucleic acid. And the basic thing that makes up the chromatin material are the histone proteins, which are the basic materials. And the histone proteins comprise about 60% of the chromatin, and the DNA or the nucleic acid, which is the acidic component, that comprises 40% of the chromatin material. Okay. Now we have to see that how this DNA is coiled within this, the acidic DNA is coiled on the basic proteins or histones to form a thread-like structure which we know as chromatin. So what happens is that this histones, you have a set of four subunits in the histone proteins. Now they are called basic proteins because they are basically made up of two basic amino acids, which are lysine and arginine. L-Y-S-I-N. Arginine. So these are the two basic amino acids that make up my histone proteins. Now we see that the histone proteins, you can imagine them like a football which has got 8 small balls inside, four small balls inside it. Okay. Now these histone proteins are in the form of subunits, which we call as, first is the H1, then second is the H2A, H2B, H3 and H4. Now this H2A, H2B, H3 and H4, they kind of form an octamer of two subunits each. H2A and H2B, H3 and H4, they will form two subunits each to give form or give rise to an octamer. That is a subunit having 8 units, small units inside it. A unit comprising of 8 small units inside it, which is formed by the H2A, H2B, H3 and H4 subunits. Okay, and they are all joined, and together what will happen is that this octamer of the subunits will have DNA wrapped around itself. This octamer is a basic protein. Okay, it's a positively charged unit, and over this positively charged basic protein, you will see the acidic DNA getting itself wrapped over it. Now, since the DNA is negatively charged and the basic proteins are positively charged, so they will tightly wrap themselves over this, means the DNA will tightly wrap itself over the subunits H2A, H2B, H3 and H4 to form an octamer. Okay. So let us read what is given in the third point. The coiled structure of DNA is held together by a protein of 8 histone molecules. DNA wound around the histone proteins to form a nucleosome. So when I have DNA getting wrapped itself on this subunit or these walls, this is called as, or it forms a structure which is called as nucleosome. And this nucleosome will have how many base pairs present in it? It will have around 200 base pairs. Now, what are base pairs that we would be seeing when I do the structure of DNA in detail? So it has approximately 200 base pairs of genes, 200 base pairs DNA wrapped around the histone protein. Now the entire chromatin structure fiber is coiled and supercoiled, something like the coils and supercoils, as you see in a typical telephone cord. So it is here I've shown a picture of a carpet which is being rolled, and after it is being rolled, it forms a rod kind of a structure. Yes. So here you have a thread-like structure. This thread-like structure is forming huge supercoils, which you see in a telephone wire. Now, how will the wire get supercoiled? It needs a structure. It needs a base to support itself, isn't it? So you can see here these histone proteins, they are forming the structure or the base over which the DNA thread is getting itself wrapped. It's just like your guddi, your latai, which you fly, or the kites which you fly during Makar Sankranti. You have a spindle, एक लटाई होती है, लटाई जिसके ऊपर तुम्हारा मांझा धागा होता है. You cannot hold the manja dhaga in your hand and fly your kite. Excuse me for my voice, children, today. So what happens is that you require a spindle. एक चाहिए तुम्हें लटाई जिसके ऊपर तुम वो मांझा धागा लपेट सको. अगर वो स्पिंडल लटाई नहीं है तो फिर इतना सारा बल्क और वॉल्यूम ऑफ थ्रेड आप मैनेज नहीं कर पाओगे in order to fly a kite properly. So here your latai is forming the structure or the base to hold on the manja thread or that thread जिससे कि तुम अपनी गुड्डी को या काइट को बांधते हो. Yes, to fly in the sky. तो वैसे ये जो स्ट्रक्चर या बेस का जो काम कर रहा है हिस्टोन प्रोटीन, जिसके ऊपर ये क्रोमैटिन डीएनए के धागे लिपटे हुए हैं. ओके, सो इन दिस डायग्राम these are the subunits which are histone, and over here you can see they are wrapping itself, the DNA is wrapping itself on the nucleosome, forming a nucleosome on this octamer of histone proteins. ठीक है, और एक कितना टर्न लगा रहा है? When one nucleosome is formed, then how many turns of DNA on the chromatin is wrapped over it? It is around 1 and 3/4 of the length. Okay. So it appears, और एक बार जब वो बंध जाते हैं तो how do they look like? They look like beads on a string. जैसे मोती रहते हैं, तुम्हारे मोती की माला होती है, तो वो वैसे मोती जैसे लगते हैं on a माला, on a string. So let us just give a quick reading. Our nucleic acids are of two types, DNA and RNA, that is deoxyribonucleic and ribonucleic acid. Yes, and this chromatin fiber, it is composed of biomolecules called as nucleic acids, and of course proteins which are called as histones, जो कि basic होते हैं because they are made up of lysine and arginine amino acids which are basic in nature. So this is just another illustration to show you how the entire, हमने बोला था कि the entire, if I take the entire length of the chromatin which is present in one body which is made up of 40 to 50 trillion cells, तो उसमें जो जितना DNA होगा that length would be more than 300 times the length between sun and earth. तो जैसा आप समझो कि इतना DNA है, तो इसको कॉइल करना है विद इन अ स्ट्रक्चर कॉल्ड सेल, जिसका की डायमीटर तुम्हारा माइक्रोमीटर्स में रेंज कर रहा है, मतलब 1 मीटर का 1 मिलियन टाइम स्मॉल साइज के रेंज में कर रहा है. उसके अंदर मुझे पैक करना है उस थ्रेड को which is 2.2 meters long in each cell. तो you can see this silver beads, ये क्या है? हिस्टोन प्रोटीन, जिसके ऊपर ये क्रोमैटिन धागे ऐसे लिपटे रहते हैं और ऐसे लिपट के ये बहुत ही हाईली कंडेंस्ड स्ट्रक्चर बनाते हैं. ठीक है, और लिपटने के लिए एक स्ट्रक्चर और एक बेस का काम, बेस प्रोवाइड करता है हिस्टोन प्रोटीन्स. Yes, which are basic in nature, over which the acidic DNA itself gets wrapped, जो कि 1 and 3/4 terms wrapping होती है, and together this complex formed between DNA and the histone protein जब एक दूसरे के ऊपर लिपटे हो जाता है तो इनको बोलते हैं हम nucleosomes. So nucleosome is nothing but a complex of DNA and the histone proteins. Right? Now, in the next structure, I will also explain you this telomeres of the end parts of the chromosomes. ये क्या है? Centromere, the highly condensed structure, and this is the arm. This is the condensed chromosomes. This was just another diagram to give you a more clearer view as to how the condensation or packing of the chromosome is taking place within the cell. And I also told you that I would be giving you another diagram to show you or help you understand what one chromatid is. So this was one chromosome. When it gets duplicated, it becomes like this. It is still one chromosome because it is still having one centromere in it. But this one half is called one chromatid, and this is another chromatid, which you can see in this particular diagram. So this yellow part, the two yellow parts which you see, they depict the two halves of the chromosome, each one half representing one chromatid. The ends are called as, of the ends of chromosome are as telomeres. The central portion which holds the arms of the chromosome is called centromere. Okay, and this entire structure, this entire X-shaped structure which you see is nothing but your chromosome. Okay, so this is your chromosome. Fine. But let us just give a reading. Chromatid is one half of the duplicated chromosome. Before replication, one chromosome is composed in only one DNA molecule. But after replication literally means, children, duplication. It literally means duplication. So in replication, one DNA molecule is copied and the two molecules are called as chromatids. During the later stages of cell division, these chromatids, they separate longitudinally to become individual chromosomes. So that we'll see later on. And what is centromere? We already know that is the constricted point at which the two chromatids forming the chromosomes are joined together. Now, little bit more clarity on chromosomes. Now, we all know that chromosomes are the hereditary material which get passed on from one generation to the next generation. They are the bearers of the heredity, bearers of the characters from the parents to the offsprings. So in human beings, how many chromosomes are present? Human beings, 46 chromosomes are present in 23 pairs. Yes? And those 23 pairs, the first 22 pairs are called as autosomes, and the last pair is called as the sex chromosome. So those 23 pairs which make up the 46 chromosomes present in human beings, the first 22 pairs are called as autosomes and the last pair is called sex chromosome. So in this particular diagram which you see, children, you can see that there are 23 pairs present. So you can see that they are numbered as well. This is the first pair, second pair, third pair, fourth pair, and so on. And this is the 22nd pair. So till here, the chromosomes are called as autosomes, and the last pair is called as the sex chromosome. Now, why the chromosomes are in pairs? The chromosomes are in pair because each part of the pair has come from one parent. Because the individual has got the heritable characters from both its parents, the mother as well as the father. So each part of the pair is contributed by mother and the other part is contributed by the father. So that means this part, if this is one, this is the first chromosome, the first, maybe this part may have come from father. The first pair of the first pair, the first part of the pair may come from father and the next one will come from the, and together they will make as one pair. So the hereditary characters are always in pairs and that pair has been formed by both the parents, mother and father. One part of the pair coming from the father and the other part coming from the mother. So now take a look at the last pair, or this is the 23rd pair. 23rd pair, which is called as the sex chromosome. In this particular diagram, you can see that it is made up of X and Y chromosomes. So this X chromosome must have come from mother. Why it has come from mother and not father? Because females or mothers have got X and X chromosomes in them, and father has got X and Y chromosomes. So this particular individual who has the one part of the 23rd pair as Y chromosome, then that Y chromosome must certainly be coming from the father. Okay. So this has come from the father, and together they form the 23rd pair as X and Y. So if X and Y is the combination of chromosomes in the 23rd pair, then the individual born is a boy. And if X and X is the pair of combination in the 23rd chromosome, then the individual is a boy. So this particular chromosome pairs which you see are that of a boy, because the 23rd pair contains one X and one Y chromosome which has come from the father, giving the sex of the child to be a male child. Now, depending upon the location of the centromere also, we can classify the chromosomes. So we take a look at from the last diagram, children, take a look from this last diagram, which is the biggest diagram. This is the metacentric chromosome, wherein the centromere is exactly in the central part of the chromosome. That is, meta stands for center. What does meta stands for? It stands for center. So you can see that the centromere is present in the center position or the middle position in this chromosome, so that both the arms, the P arm and the Q arm, which you see, the P arm and the PQ arm are almost of the same length. So when such is the state of the chromosome, such that the P and the Q arms are almost identical, then it is called as a metacentric chromosome. And when metacentric is the chromosome, then the shape of the chromosome becomes V-shaped. Child, here the shape of the chromosome will become V-shaped. So you may be asked a question like V-shaped chromosome, then it represents metacentric. For submetacentric, it either you see it becoming L-shaped or J-shaped. Why? Because the centromere position is now shifted from the central position. It has moved a little bit away from the central position, such that the P arm is not completely equal to the Q arm. Okay, the P arm is a little bit shorter than the Q arm. The centromere is not exactly in the center. It is slightly moving away from the center, giving this chromosome L-shaped or J-shaped. So you can get questions related to the shape that is belonging to which type of chromosome. Then you will have to answer it be a submetacentric chromosome if it is L-shaped or it is J-shaped. And then you have the acrocentric chromosome, where you have the centromere almost present at the one end of the chromosome, such that you have got one arm being very big than the other arm. That is, a Q arm becoming very, very big as compared to P arm. There is a lot of difference between the lengths of P and Q arms. Such is called as the acrocentric. We don't find them in our body. Telocentric are not found in our body, in which you see that there is hardly any P arm left. Okay, because the centromere has moved on to only one end, completely lying towards one end of the chromosome. When the centromere is placed, then it is called as a telocentric chromosome. Okay, we do not find this in human beings. So that's where the centromere is at the proximal end, that is at the far end. So this is about the classification of the chromosomes, children. You can give a reading. Telocentric chromosomes are rod-like chromosomes with centromere present on the proximal end. There is no P arm. So you can get a question, the type of the chromosome in which you do not see any P arm. So that is called as a telocentric chromosome. Okay, because they are not found in humans. Acrocentric means a very, very small P arm and a very long Q arm. Okay. Then submetacentric, it will be L-shaped or G-shaped, which with centromere near the center of the chromosome, giving rise to two unequal arms. So you can get a question related to a chromosome in which the arms are unequal and it gives rise to L or J shape. Then you have to give the answer that it will be submetacentric. Then metacentric, when it is completely in the center, giving rise to a V-shaped structure, such that the P arm and the Q arms are equal in length with each other. Now coming to the next very important topic called as the homologous chromosomes. Now you will come across this word homologous chromosomes very much when we are doing in detail cell division as well as genetics. What are homologous chromosomes? Homo means alike, similar. Okay, so now you know that chromosomes are always present in pair. So if I talk about my sixth chromosome, then it will be in pair. Why will it be in pair? Because my sixth chromosome is made up of one part of the sixth pair of the sixth chromosome coming from my father and one part of it coming from my mother. Okay, one part of the sixth chromosome coming from my father and one part of the sixth chromosome coming from my mother, right? So my sixth chromosome is having a pair. But which are the two pairs? Those two pairs, how will they be identified? And they will be called as homologous chromosomes. They will be identified to each other in the sense that they will both be having similar shape and size. Both the pairs, now you can see here, even though the colors are different, one is green and the other is pink in color, but the size, shape is similar. Yes, and the genes which would be present on the chromosome, suppose this gene A is present on my one homologous chromosomes of the sixth pair. And this is the next, this is the gene A dash, which is present on the other pair of my sixth chromosome. So what will I see is that both A and A dash would be contributing for the same traits within me. Like suppose it is for tallness in me. Then maybe A will contribute for tall height and A dash will contribute for short height, which has maybe come from one parent and the other has come from the other parent, mother and father. So I can say that A and A dash, they are basically the same genes, but the traits are different. That is, one is for shortness and the other is for dwarfness. Tallness, and one is for shortness. So the gene is the same. So when I see that the shape and the size of the chromosomes are similar to each other, and they both have the same types of genes present over them, genes for the same features, that is the feature height in this particular case. Both gene A and A dash are responsible for determining the height. Now they may have variations. One may be for tallness, one may be for shortness, but both are for height. So, and the location of the gene for the height is also same in both the chromosomes. So these two chromosomes are not just similar in shape and size, they're not just similar in the type of gene being present, but also the location of the gene present on both these are also same. So such pairs are called as homologous chromosomes. So let me give a reading. It refers to a pair of chromosomes having the same gene sequences, each derived from one parent. So I say that one maybe has come from mother and this part coming from the father. They will all have the same shape and size. Both these will have same shape and size. Okay. And they might have very slight differences. So I'd say that they are not identical. They are not completely identical. They will be similar in shape and size. There is a difference between the words similar and identical. Identical will be a Xerox copy. So it's not a Xerox copy. You can see that the genes are different here. One has a gene A and the other has a gene A dash. Like one A may be representing a very tall height and A dash may be representing an average height. Okay, so that's what is the meaning here. They contain slight differences in their genetic information, allowing each gamete to have a unique genetic meaning. So you will understand this third point more when we do a meiosis, particularly in detail. That is how it results in variation. How these slight differences give rise to so much of variations which we have between us and our parents, between us and our brothers and sisters. Now that we know that what the chromatin material is made up of, it is made up of DNA and the histone proteins. So let us see what DNA and RNA are made up of. What these nucleic acids, DNA and RNA, are composed of. So DNA and RNA, they are polymeric molecules. They are polynucleotides. What is the meaning of polynucleotide or a polymer? I hope you know the meaning of the word polymer. Polymer is when one unit is repeated again and again, it forms a polymer. So when monomers means the same units, like suppose if I have one molecule here A, it is repeating itself again and again, then it becomes a polymer. Wherein, what does A represents? A represents a monomer here. So similarly, DNA is a polynucleotide. So it must be made up of, what is the monomer here? Monomer is the nucleotide. The monomer is the nucleotide. So nucleotide, many units of nucleotides, so many repeating units of nucleotides forms the polynucleotide. Giving now, and what is this nucleotide comprised of? Now you must be looking at the structure, children. Now you can see this letter A, T, and these balls here. So that means what? This nucleotide is made up of many things. So yes, this nucleotide, let me say that this nucleotide is made up of 3 parts, A, B, and C. Okay. Now this A, it represents phosphate. Okay. What is phosphate? PO4, 3 minus radicals, charge of 3 minus. Then what does this B represent? B represents a sugar which is called deoxyribose sugar. Now sugars are what? They are carbohydrate molecules. They may be of various types. They may be made up of 3 carbon atom. They are called as trioses, 4 carbon atom tetroses, 5 carbon atom pentoses, 6 carbon atom hexoses, heptoses, and so on. So the DNA particularly is made up of a deoxyribose sugar. What is this deoxyribose sugar? Deoxyribose sugar is an example of 5 carbon sugar compound. So it's a sugar compound which is made up of 5 carbon atoms in it. So if this is the oxygen atom, something like this. So 1, 2, 3, 4, and the 5th carbon is here. So this is somewhat like I have given a condensed structure of the sugar molecule. It is a deoxyribose sugar, which is a pentose sugar, which forms the second part of the nucleotide. And what is the third part of the nucleotide? C, that is nitrogenous base. But children, what is a base? You have studied your chapter acids and bases, which is the 4th chapter in chemistry. So acids are something which have replaceable hydrogen ions when you dissolve them in water as the only positively charged ions. And bases are those substances which have got replaceable OH minus ions in them as the only negatively charged ions in them. So nitrogenous bases are those bases which will have nitrogen present in them and will have OH minus as the only negatively charged ions also present in them. So they will give you all the properties of base. They will show you the properties of base. So they are called as bases and they also have nitrogen in them. So these 3 things make up one monomer or one nucleotide of the DNA. What makes one nucleotide of DNA? Once again, a phosphate. Child, look here, a phosphate. Then a sugar, which is deoxyribose sugar. And third part is a base, which has nitrogen in it. So it's a nitrogenous base. Now take a look at this particular structure. So this structure, it represents kind of a spiral. It represents a kind of a spiral staircase. जैसे कि एक पुराने घरों में bungalows में दिखता है. So I can say that my DNA is a double helical structure. Double helical structure. That is, it is made up of 2 helices. You can see this is the one strand and this is the another strand. Okay, they are coiled. Okay, they are coiled and giving it this kind of a structure. Okay. And as I said, that the DNA is made up of nitrogenous bases. The nitrogenous bases are of 4 types which you see in the DNA. Nitrogenous bases are of 4 types which you see in the DNA. I will classify these 4 types into 2 first. They are purines and pyrimidines. Okay. The nitrogenous bases are first classified into purines and pyrimidines. Now, purines are double ring structure. They are made up of 2 rings in them, and the examples are adenine and guanine. Adenine and guanine. And pyrimidines are made up of single ring structure, and the examples of pyrimidine are cytosine and thymine. So, together they make 4 nitrogenous bases. So, the nitrogenous bases are adenine, guanine. Adenine was represented with the alphabet A, guanine G, then cytosine C and thymine T. And you see that adenine always pairs with thymine with hydrogen bonds in them. So A pairs with T and G will pair, that is guanine will pair with cytosine with 3 hydrogen bonds. Fine. So you can see here that this green is cytosine. This is one nitrogenous base, and this will pair with G, that is guanine, with the help of 3 hydrogen bonds. Can you see 3 hydrogen bonds here? 3 lines, 1, 2, 3. So these 3 lines represents 3 hydrogen bonds which are present between the nitrogenous bases, cytosine and guanine. And adenine and thymine, you can see that adenine and thymine are held with each other by means of 2 bonds. The details of this particular structure, you don't have to do it in grade 10. This is just to give you that what are the components that make up the DNA. In grade 10, you should know the DNA is made up of nucleotide having 3 molecules. One is a phosphate, one is a deoxyribose sugar, which is pentose, that is made up of 5 carbon atoms. And the third part is made up of nitrogenous base, which can be a purine or a pyrimidine, that is either adenine, guanine, cytosine or thymine. And they will be repeated, and these 3 will be repeated over one another. This forms one nucleotide. And when it is repeated again and again, it forms a polynucleotide. So thus our DNA is a macromolecule or a polynucleotide, where we have got repeating units of phosphate, sugar and base with each other. So you can see here phosphate, sugar and base. This particular diagram, take a look at my pen as it is moving on this diagram, children. So the blue color is for the phosphate molecules. This gray is for the sugar, deoxyribose sugar, and the purple color or the red colors and yellow and green are for the nitrogenous bases. So this comprises one nucleotide. Then this is another nucleotide. This is another nucleotide. And they are bonded to each other. They are all joined with each other in the form of long chains, giving rise to a double helical structure of the DNA. What is the full form of DNA? Deoxyribonucleic acid. What else does the DNA do? It controls the overall metabolism of the cell. Why? The DNA has the ability to control the entire cell. So the entire machinery of the cell is actually controlled by the DNA because it has got codes in it, because it has got information on it which will be making proteins, and proteins would be doing all the work that is necessary to be done. So to carry out all the work of the cell, where is the necessary information present? All the necessary information is present within the DNA. So thus I can say that DNA controls the overall metabolism. What is metabolism, children? Metabolism is the sum total of all the chemical reactions, whether it is catabolic reactions, which are called as catabolism, or it is the anabolic reactions. So any kind of reaction, the information to control those reactions is present within the DNA. Transcription, that is the formation of proteins, which you don't have to study in detail, but you should not be unaware of the fact of what should be the meaning of this word transcription. So when the transcription word is uttered, you should know that it is protein synthesis. It is the making up of proteins by the codes or by the messages which are present in the DNA. Okay. The next is, of course, we all know this transferring the genetic information from one cell to its daughters and from one generation to the next is also done by the DNA. DNA is also responsible for the evolution. What is evolution? It is the change in the characters of an individual getting passed on from one generation to the next generation. So it is the changes which you see between one generation to the next generation in an offspring or in a species or in a population. It is called as evolution. So who is responsible for this evolution? It is the DNA which is responsible for this changes in the characters between one generation and the next generation of a species or a population. So that also is brought about by cell division with the help of the DNA. So this also is one of the functions of DNA. Next is DNA fingerprinting. Now DNA, what is DNA fingerprinting? Now this is a very fashionable word these days because it's used a lot in solving paternity issues, in finding out criminals, and a lot of such kind of cases. and who was present at the site of the murder, and kinds of DNA fingerprinting helps to give the genetic makeup of the individual. It is a kind of a mechanism process which deciphers or gives you the entire genetic makeup of an individual, which actually helps in what? Which actually helps in knowing the paternity issues or solving the cases of many criminal cases. Okay. Next comes, what are genes? We already have studied what genes are. They are nothing but the specific. Now we said that the DNA is 2.2 meters length in each cell. So does this 2.2 meter entire length, does it have genes over it? No. There are only some sections which will have information on the DNA, which will be able to make up a protein, or that meaning to say that there will be only some locations on the DNA which will have informations which can be processed. That information can be processed to make up a protein. So such segments on the DNA which have information on them to form proteins are called as genes. Okay, it will be more clear in this particular diagram. So this red colored chromosome has got silver stretches on it. Can you see this silver stretch here, silver stretch here? So this is genes. When you open this silver stretch, it has got DNA. No, genes are nothing but stretches of DNA which will have information on them. This is the information which will give rise to, or which will form a protein which will do the chemical reactions of the cell, isn't it? So those segments on the length of the DNA which are able to code for a particular feature or character or protein are called as genes. So let me give a reading. Genes are the specific sequences of nucleotides on our chromosomes which encode, or that is contain information for giving reactions or making proteins which express in the form of some particular feature on the body, like the feature of my color. If this is the color of my skin, so for this color of the skin, there should be a particular protein made. So for making that protein, I should have the code to make that protein. So that code is present in my gene. So the length of the DNA where that particular gene is present, which was able to make the protein responsible for my skin. So that is called gene. So similarly, for all the features of the body, those specific lengths of the DNA containing information for that particular feature are called as genes. Excuse me. So genes are nothing but the molecular units of heredity. In this particular diagram, children, you can see that this is loci, means location. So this location is for a gene for eye color. So this gene, this place has got certain nucleotides, A, G, C, T combination of bases, which are giving the characters, which are able to prepare a protein which can give the color to my eyes. Okay, and at this particular place, you can see pink color. This section contains nucleotides or the genetic information which can make proteins which can give color to my hair. Okay, so the color to my hair is given by a protein which is made with the help of this sequence which is present on this zone. So this functional unit or this length of the chromosome is called a gene, which is having information to code for the protein which can give color to my hair or color to my eyes. Okay, understood? I hope so. Now coming to the second nucleic acid which is present in our body, and which is RNA. What is it, children? It is RNA. RNA, what is the full form? Ribonucleic acid. So this is another nucleic acid which is present in all the cells. Now, unlike DNA, which is present only inside the nucleus, DNA is present only inside the nucleus of the cell, and we have also studied that it is present inside the mitochondria, and it is also present inside the chloroplast. So other than these organelles, you will not find DNA in the cytoplasm, whereas ribonucleic acid RNA is present also within the cytoplasm of the cell. Okay, so unlike DNA, it is not double coiled, it is only singly coiled. We saw that DNA was double coiled structure. It had got 2 strands, but RNA has only one strand. has got only one strand. So just like DNA, RNA is also made up of units called nucleotides. That is, it is also made up of repeating units of nucleotides. Nucleotides which will be made up of one phosphate molecule, one sugar molecule, and one and nitrogenous base. So let us see what is the phosphate will be the same. Phosphate is always PO4 3 minus. Let us find out whether the sugar molecule is also the same and whether the nitrogenous bases also are same in RNA from DNA. So we see that the sugar molecule which makes up RNA is not deoxyribose, rather it is ribose sugar. So the name, it is also of course a pentose sugar. Ribose and deoxyribose, both are examples of pentose, that is the sugar having 5 carbon atoms in it. But DNA contains deoxyribose sugar, that is, it does not have one oxygen atom in it, as compared to RNA, which is a pentose sugar, which is called as a ribose sugar. You do not have to do the structure of the deoxyribose sugar and ribose sugar, which is why I'm not giving you those structures here. They are there for you in grade 12. So the sugar is different. DNA you had deoxyribose, RNA you've got only ribose sugar. And what are the bases? The bases present, you can see that the bases here, adenine, guanine, cytosine are the same, but thymine is not present. Instead of thymine, what do you see? You see another base which is called as uracil. Okay. So one of the purine bases in the DNA, that is thymine, that is replaced by uracil in the, okay, so instead of thymine, you will have uracil, the nitrogenous base present. RNA is single-stranded molecule because our DNA was double-stranded. RNA is present both within the nucleus as well as outside the nucleus, that is inside the cytoplasm. And we saw that DNA was present only inside the nucleus of the cell and on the mitochondria, chloroplast. Okay, it was not present inside the cytoplasm. Let us give a reading to the functions of the RNA, children. What does the RNA do? It facilitates the translation of DNA into proteins. That is how the information of the DNA is passed on. It is passed on by making proteins. And in the process of making of proteins, it is the RNA which is very much helpful, because DNA is not going to leave the nucleus. It contains all the information for cellular mechanism, for the cellular reactions, but it will not leave the nucleus. So how will it pass on the information? There has to be someone who can come out of the nucleus, can carry the information of the DNA and come out of the nucleus and pass on the information to make the necessary proteins for particular features or reactions. So that role of carrying the message from the DNA and making up of proteins is done by RNA. So therefore, RNA has a very, very important role in the formation of proteins, that is passing on the information of the DNA to be in the actual process, that is the actual information, how it is that role is fulfilled by making up of proteins, and which is done by RNA. Then RNA also functions as an adapter, that is carrier molecule in the protein synthesis, serves as a messenger between DNA and ribosomes, and it is the carrier of genetic information in the living cells. Yes, it is carrying all the information of the DNA. Okay, children? So we did try to cover some of the important terms that we would be constantly using while we are doing the chapter genetics and cell metabolism. And also, since we are biology students, even though only of grade 10 now, and there are a lot of things which we do not have to take up at this grade, but still it is important as biology students that we are able to understand the terms in clearly as to what the word chromatin means, what chromosomes are, what chromatid is, and what is a centromere. So in this particular class, I have tried to explain you the slight differences which you see in not just the spellings of these 4 words, but also the slight differences that is present between what chromatin and what chromosomes are and what chromatid. It's just half of the chromosome, isn't it? Centromere, the central position. Yes, then we also saw a little bit about the structure and the function of DNA, and also the structure and the function of the RNA. So I hope this particular class was able to solve some of your queries related to these very similar sounding terms such as chromosomes, chromatin, chromatid, centromere, etc. In the coming class, that is in the next class, I would be beginning with cell division. So that is all for this particular session. Please go through the session so that you are able to revise this particular concept, and this unwinds the first part, that is cell and the basic terminology. Then we will be doing mitosis. First of all, we will be studying as to why cell division is important, and then in length we will be seeing how cell division actually takes place in the next class. So hope you're enjoying my session. Keep watching my videos so that you get more and more clarity for your board examination. Thank you very much, children. Bye-bye.